welcome back. We have been discussing about the basics of nanotechnology, and in that context we have talked about the different type of geometry of nanomaterials. Followed by that we have talked about the synthesis, and in the synthesis we have talked about the physical synthesis followed by the chemical synthesis, and now we will proceed towards the biological synthesis. As I told you in the initial outline that biological synthesis is one of the most smartest synthesis in terms of conserving energy and in terms of conducting the reaction at a very low temperature or a pressure or within the physiological regime. So, there is a lot more to learn about the biological synthesis of nanomaterials, because that is where the future lies, a very sustainable route. So, if we recollect what we talked about the biological methods, so basic idea is will be emulating nature, biosynthesis which is a green route, and the raw materials are in friendly and these are eco-friendly things, and understanding the nanoreactors or nanovessels where in a confined space the enzymes execute the addition and subtraction of bonds and leading to a self assembly. Okay. And while we talk about the biological route, these are mostly environmentally benign, low toxic, cost effective and more efficient modules. So, the three routes what has been followed as of now pretty much well documented are microorganism assisted biogenesis, biotemplate assisted biogenesis and plant extract assisted biogenesis. So, we will start our journey from where out here, and all these techniques employ either biological systems like bacteria, fungi, virus, yeast and actinomycetes and plant extracts. Okay. Let us resume from here. Biological route of nanomaterial synthesis. And the biological route of nanomaterial synthesis, as we have already mentioned, the first one we will be discussing is biogenic synthesis using microorganisms. Biogenic synthesis using microbes or microorganisms. So, the basic idea of biogenic synthesis using microbes is prokaryotic bacteria like actinomycetes, fungi, algae, yeast. Okay. Prokaryotic bacteria, fungi, yeast, actinomycetes, they are extensively used as bioreactors. So, these functions as the bioreactors. If you consider these single cell organelles as single cell organisms as bioreactors, and these bioreactors are used to synthesize the nanomaterials. And if you look at it, the enormous scientific effort were made to develop a strategy of producing nanoparticles of silver, gold, palladium, titanium, cadmium, mostly cadmium sulfide. Okay. Microorganism grab the ion targets. The way they do it is very straightforward. So, these microbes basically they grab the target ions from their environment and they turn the metal ions into element metal through enzyme generated by cellular activities. Okay. The synthesis can be classified into intracellular and extracellular. So, for example, they are pulling the ions. So, this is the environment outside. So, if this is the microbes, and this is the surrounding environment. They are pulling the raw material from the environment. It could be X, Y, Z, whatever. And out here, by enzymatic reaction at a very low temperature and pressure, they are converting them into some of the intermediate whatever we are asking for, and then this is the time where we are intervening into the system to extract out these desired products out of through purification and different kind of routes. Okay. Now, this is what is about the basic outline of the process, and but the synthesis could be classified into two levels. This could happen either, so for example, I'll follow a different route strategy. It can happen at this level also, at the surface. Okay. So, in that situation, what we are having is the microbe is say decorated with a series of enzyme on top. So, these what I am drawing now are the decorated enzymes sitting on top of the microbes, and these are helping in transforming some X, Y, Z, whatever, into some desired product by the enzyme activity, to say XA, XB, XC, or likewise, okay, depending on what is the desired product we are asking about. But depending on where it is happening, you have either these are extracellular or these are intracellular. If it is happening outside like this, we call this kind of synthesis as extracellular synthesis. And if it is happening inside the cell, here we call them intracellular synthesis. These are the two modes of synthesis following the microbes. The intracellular method involved transporting the metal into the microbial cell to form nanomaterial in the presence of enzyme, as you could see. So, the transport has to happen, so which is slightly more energy intensive. The extracellular synthesis of nanomaterial involved trapping the metal ion on the surface, as you could see, on the surface of the cell, and reducing the ion in the presence of enzyme. So, bacteria utilize number of anionic functional groups, proteins and enzyme reducing sugar in bacterial biomass to reduce interacting metal ions. Okay. So, this is the basic approach, and this could be done by fungus, where fungus mediated green chemistry approach for synthesis of nanomaterial has several advantage in terms of bioaccumulation, economic viability, scaling up synthesis due to simple downstream processing and biomass handling. Okay. That suppose you are using fungus, so most of these processes are fairly economical, and this is the part what we are talking about, the downstream processing, where you are isolating the desired product at a particular point, and much of these downstream processing are well within the economic limits of producing such nanomaterial. So, this is one of the basic methods in biological synthesis. The next one is, and this line will be coming to template assisted, which is biomolecule as template to design nanomaterial. Biomolecules as template to design nano materials. Okay. Now, when we talk about template assisted synthesis, so there are various biomolecules like you can use nucleic acids, you can use bilipid membranes, you can use different viruses, you can use diatoms. Might wonder what are diatoms. These are algae, these are single cell algae, and these are really one of the most populated algae in the algae family, and they have a silicate cell membrane, and those are really beautiful silicate cell membranes. If you just give a search in Google Images and you go for diatoms silicate covering, you will see amazing patterns in them. So, these diatoms could be used as a very interesting template for any kind of lithographical applications, and as a matter of fact, there are groups in the world who are trying to use those kind of pattern templates for bioelectronics application, for developing transistors, developing electronic, using them as electronic substrates. Okay, coming back where we were. So, you have nucleic acids, membranes, viruses and the diatoms. These are used as template to synthesize nanomaterial. DNA is widely known as an excellent biomolecular template that has a strong attraction with transition metal ions. For any transition metal ion nanomaterial synthesis, DNA could be used as a template. It is shown that the DNA hydrogel could be made and cross-linked before incorporating transition metal ions like gold to DNA macromolecules that eventually lead to the formation of gold nanomaterial. The process involved reduction of gold from its 3-state leading to the formation of gold atoms and metal cluster that developed into gold nanomaterial on the chain of DNA. So, this is one way you can use DNA, and DNA mediated synthesis or strategy to synthesize highly stable wire like clusters of silver nanomaterial is also a very commonly observed area of research. Okay. So, one such area, so basically the whole idea is you are using a template. In this case, the template is nucleic acid. Similarly, you can use diatom as a template. So, on this template you are attracting your material which you are going to convert on this template to something like this, as I was showing in the previous example. Something like this. Okay. So, these are template assisted synthesis, and in the third in that series is the next one, which is plant extract for nanomaterial synthesis. Plant extract for nanomaterial synthesis. Okay. So, the biosynthesis of nanomaterial using plant extract or plant biomass is one of the very effective, rapid, clean and non-toxic and eco-friendly method. This method has been utilized predominantly to synthesize nanomaterials of noble metals, metal oxide, bimetallic alloys, and they have been adequately demarcated various plant biometabolites that could help in preparation of nanomaterial based on their valuable role as reducing agent and as capping agent. One of the very common logics what is being followed in this kind of plant extract based nanomaterial synthesis is you are utilizing the specific enzyme needed for forming some specific bond which otherwise in an inorganic setup could consume significant amount of energy and power. So, you are in a way you are manipulating the milieu of the reaction by introducing an enzyme, and these are some of the very clean methods, and these are economical and they have a simple downstream processing, and they are free from much hazards because you are not now dealing with living microbes, you are just using a plant extract. So, the core idea is very straightforward, as I mentioned. For making or breaking any kind of bonds, the nature uses series of enzymes. As a matter of fact, our story of life could also be called as story of enzymes. So, here we are exploiting those gifts of nature to us, those wonderful enzymes to make or break bonds to synthesize the desired product or desired nanomaterial what we wish to obtain. So, let us summarize what all we have covered in this section as of now. To start off with, so this is where we started on the module of nanotechnology. We talked about the classification of nanomaterial in terms of shape and geometry and the chemical nature. There we talked about nano cages, nanocrystals, nanobelts, nanofibers, nanoparticles, nanotubes, both hollow and solid, nanowires, quantum dots and nanocomposites. Okay. Then in terms of synthesis of nanomaterials, in terms of the chemical nature, they could be either organic, inorganic or carbon based, and in terms of the synthesis, we could have either top down approach or bottom up approach, and these are further classified based on the route we are following. They could be physical, chemical or biological. Physical, we have talked about all the techniques: high energy ball milling, pulse vapor deposition, laser pyrolysis, flash spray pyrolysis, electrospraying, melt mixing and polymer composite development. Whereas in the chemical section, we talked about sol gel synthesis, microemulsion technique, hydrothermal synthesis, polyol synthesis using polyethylene glycol, chemical vapor synthesis, plasma enhanced chemical vapor deposition. And now in the biological segment, we talked about microorganism assisted biogenesis, biotemplate assisted biogenesis and plant extract assisted biogenesis. So, in this segment, now we are left with the characterization of nanomaterials. So, this is in the module of nanotechnology. This is where we will be concluding once we talked about the characterization. So, we will close in here, and in the next class we will talk about the characterization of these different nanomaterials, characterizing them based on their geometry, characterizing their chemical nature, characterizing their specific functional groups which are available, and that way we will be concluding this section. and the way we are moving. So, your first week we have devoted about the role of agrochemicals in agriculture and the need of nanotechnology in terms of precision farming, in terms of reducing the usage of optimizing, rather the right word will be optimizing the use of fertilizers and pesticides. From there, in the week 2 we moved into the nanotechnology, the basics of nanotechnology, and there is a slight spillover of the second week into the third week. And after characterization, so we talked about the classification of nanomaterial, we talked about the synthesis, and now we will be talking about the characterization. After characterization, we will move on to specific nanomaterials which have been tried out in the laboratory, in the field for agricultural applications. Okay. So, let us catch up in the next class with the characterization of the nanomaterials before we proceed into the application of nanomaterials in agriculture, which includes plant, animal production, food packaging or post harvest technology and cleaning of water. Thank you.